Okay, we're moving on to the skeletal system. Let me get this screen shared. There we go. Okay. So the skeletal system functions. It functions in support. It provides a framework for soft tissues and muscle attachment. Protection because it surrounds vital organs like the brain, spinal cord, heart, and lungs, such as the ribs and the sternum, the vertebral column in the back. Movement. Well, bones move, but they don't move within the bone. But the muscles that attach to the bones will shorten and lengthen, and that provides movement of the skeletal system. Mineral homeostasis, restore calcium and phosphorus there. Hematopoiesis, which is a fancy word for blood cell production and red bone marrow. And then storage of triglycerides in yellow bone marrow. Right, there's red bone marrow and yellow bone marrow, and we'll talk about the difference between those. Red bone marrow can be found throughout, all the long bones and different, flat bone areas, in children. But as we become adults and reach skeletal maturity, a lot of those areas get converted from red bone marrow to yellow bone marrow. And so that's what we'll be looking at in what's called the medullary cavity. Okay, so we have, when it comes to the skeletal system, we have, and we were talking about this when we covered tissues and connective tissues. We said when it came to cartilage, we had hyaline, elastic, and fibrocartilages. So your little rhyme or mnemonic is going to be HEF. Hyaline, elastic, and fibrocartilages. On most models, hyaline cartilage areas are indicated in light blue, all right? Sometimes even a dark blue, but blue. Elastic cartilage, the areas may be in green frequently, and fibrocartilage in red. Then when it comes to the bones, we have compact bone and spongy bone. So two different types of bones, all found kind of within, but they have their matrices arranged differently and is found in different areas. So we have these three cartilages, these two types of bones, skeletal system. Okay, so here on this skeleton, it's not only using the red, blue, and green locations to indicate down in the right-hand corner, the blue for hyaline cartilage, the green for elastic cartilages, the red for fibrocartilage areas. But we also have a color depiction or differentiation between your axial skeleton and your appendicular skeleton. The axial skeleton is the skeleton, or the bones, I should say, that are found in the head, in the middle ear, your vertebral column, and the bones at the bottom of that stack, the sacrum and the coccyx, ribs and sternum. So those bones in like the darker tan, are all part of the axial skeleton. The appendicular skeleton are the rest of the bones of the arms and the legs, and the bones that attach those arms and legs, like a clavicle and a scapula, to the axial skeleton. All right. So if you know what's in the axial, then you know essentially what everything else falls in the appendicular. All right, so back to the colors. Hyaline cartilage is the most abundant of the cartilages. So here it shows you on the tip of the nose. We have costal cartilages. Costal cartilage, that's the name of the structure. It's the cartilage that attaches ribs to your sternum. The tissue, quote unquote, that we find there is hyaline cartilage. Right, we have the articular ends of bones, all these synovial joints, we call them. You will find hyaline cartilage there, and that's where the two bones come together to form a joint or an articulation, and it has a lot of cartilage there because there's a lot of movement. You can see them in the shoulder joint here, what's called the glenohumeral joint, the femoral acetabular joint, the hip joint, and throughout the feet. They're very common because it provides a lot of movement. Right? Elastic cartilage locations, ear, epiglottis, which you can't see here because it connects the back of the throat to the middle ear, but we can see them on other models, and especially when we get to the ear models of special senses at the end of the course. The ear, epiglottis, and eustachian tube. The epiglottis is found in the larynx. So up in the upper right-hand corner here, we're looking at a model of the larynx, which includes the thyroid cartilage, the cricoid cartilage, and the arytenoids. You can't see them from this direction. You have above that what's called the hyoid bone. And then you see the green little tongue. It looks like a green tongue up there, but it is a flap of elastic cartilage that closes off the airway, the entrance to the airway when you're swallowing food. So food actually goes down into your esophagus. When you need to breathe in and out, that epiglottis elevates so that it opens up the air passageway. And so we'll see that again in the respiratory center, the respiratory system as well. Right, but then you see all these other blue structures, here in that big lung model. So you, again, consistent with the light blue color for hyaline cartilage locations. We have the thyroid cartilage, the cricoid cartilage. We have parts of the trachea, the bronchi, and then that's really where it ends with the bronchi. Bronchioles and alveoli will be histologically different. Okay, so that's kind of a visual of those locations. So let's just take these one by one. Hyaline cartilage contains only collagen fibers that are not directly visible microscopically. So although there are collagen fibers there, they don't take up stain really, that we can see. So, your lab teacher may or may not consider those fibers, really part of the hyaline cartilage. I mean, it's there, but because you can't see it and you're looking at slides, sometimes they won't necessarily consider those collagen fibers part of the hyaline cartilage. But you can ask your instructor. Kind of depends on the person. But hyaline cartilage allows a lot of flexibility. It's the most abundant cartilage in the body. It's where all of our long bones originate from what's called a hyaline cartilage blob of tissue. Essentially, I'll show you what it looks like later. So it's very common. Locations in the body include the ribs, and technically the ribs are bone. We're really talking about the costal cartilage. That's the cartilage that attaches the ribs to the sternum. The trachea, larynx, nose, articular ends of bones, and the epiphyseal plate. And I need to close that parentheses. That's making me crazy. So I'm going to break that down right now. Hyaline cartilage. Close parentheses because that makes me crazy. Okay. Sorry about that. Okay, the cells found in hyaline cartilage are chondrocytes. Remember, we're talking about tissues, and we said, when it comes to the really any of the connective tissues, that the cells found there's often particular unique cells in each kind of system. So when it said it came to bone, we said we're going to see osteocytes, osteoclasts, osteoblasts. When it came to cartilage, we're going to have chondrocytes because chondro is cartilage and cyte is cell. So these are cells that form cartilage. So here's a picture of it. Now, some pictures are just better than others. I will tell you that the general, and this is fairly magnified, so generally you're not going to see it quite this close up, but you may get an example down in the lab. Here we have the chondrocytes, which is the cell sitting in a lacunae. So lacunae is like a little room that the cell can sit in, so it doesn't kind of get overwhelmed or suffocated by all the pinky purpley matrix surrounding it. If that makes sense. It's like having its own little cubicle at work, right? And so everything else is outside of the cubicle, and you can sit right at your desk, in a cubicle. Right? So the chondrocyte has a dark nucleus here in the center. You can see the borders of the lacunae where it kind of borders, it wells up some dark pink matrix around it. Well, also kind of look at how these are dispersed from one another. If we lower the magnification a little bit, you would notice that the matrix, the fluidy part here, is pretty much like devoid of seeing any kind of fibers there. But the matrix is actually rather either light pinkish or light lavender staining. So we don't really see any fibers in there. These chondrocytes are rather spread apart from one another, and it has kind of a soap bubbly appearance to it. That's what I call like a soap bubble kind of appearance. They're evenly they're not exactly evenly dispersed, but they're spread out from each other. They're not all condensed and stacked up on one another. They merge here and there, but because the way you take this slide, they're not necessarily next to each other. They might be at different levels vertically, kind of from one another. And you're looking down on the slide. Okay, so anyway, they're generally dispersed rather evenly, has a soap bubble appearance. No particular visible fibers, and often either a light pink or a light lavender background. Okay, hyaline cartilage. Elastic cartilage is different. This has very visible dark staining elastin fibers. We said elastin fibers were that medium diameter size fiber that takes up, pretty good stain, pretty dark purple stain. And depending on how dense those fibers are, that dark, that purple might look really, really dark purple, or, you might see some gooey, that light lavender kind of a matrix in between some of those fibers. But frequently you'll find areas where those fibers are stacked heavily on one another. And so the color difference between the chondrocyte in the lacunae and the fibers outside of it are really distinct from one another, I guess you could say. So it contains primarily elastin fibers and collagen, but primarily elastin. It has what we call I call it the Scooby-Doo eye appearance. Because remember Scooby-Doo, you'd always see him sitting in a closet or in the dark, and you'd have these two eyes peering out of the whole darkness. That to me is that contrast between the chondrocytes in the lacunae and the dark purple staining elastin fibers surrounding it. It is more flexible and allows the structure to maintain its shape when bending. So locations are your E locations. We have E elastic cartilage, E elastin or elastic fibers, and E locations, external ear, epiglottis, eustachian tube. Epiglottis is that part in the larynx, in the throat. The eustachian tube we'll see on some models either down in the lab or when we get to the special senses model. It connects the middle ear to the upper part of your throat called the nasopharynx. And the cells we find there are, lo and behold, chondrocytes. Right, so here we have a picture of the ear as a location. All right, so here we have a chondrocyte sitting in its lacunae. This is magnified even more than the hyaline cartilage, but you can see these dark purple fibers there, those elastic fibers. And over here, about at the tip of the line for matrix, that's generally that very dark appearance where all these fibers are packed up on one another, stacked on one another. That's usually the very dark purple appearance contrasted with the lighter staining appearance of the chondrocyte in its lacunae. And so that's what I mean. You probably think I'm crazy, but the Scooby-Doo eyes, where you can see these chondrocytes, distinctively, compared to the dark, thick, fibers, that are surrounding it. That looks very different from your hyaline cartilage. Light pinkish, light background, no visible fibers, kind of soap bubbly appearance. Okay, then our last cartilage is fibrocartilage. The cells, of course, are chondrocytes, so same kind of cells. The fibers are collagen fiber stuff, and the collagen fiber is going to take up a bluish stain, and this is and they're rather undulating. So I call it the Van Gogh, painting appearance, because you have these blue wavy lines like Starry, Starry Night. If anybody got to the Van Gogh exhibit, down at the Valley in the last couple of years, you would have seen that. They are highly compressible, and thus they have great tensile strength, which means they withstand great pressure with some flexibility. So they help withstand being compressed, crushed, crunched. So they are going to be found in areas where you have a lot of axial compression, a lot of weight loading and compression at that joint area. So intervertebral discs, the disc between each vertebra of your spine. The pubic symphysis, anteriorly where the two pubic bones of your pelvis come together. And the menisci of the knees, they're like brake pads between your femur and your tibia, behind your kneecap, your patella. So the menisci of the knees, big impact areas, fibrocartilage locations. Right, this is a slide that represents it, and this is pretty dead on with most that you'll see. Right. You have these blue wavy lines that are rather pretty, I think. Those are the collagen fibers. And when you stain like this, the nuclei of the chondrocytes have a tendency to take up a deep fuchsia color. So you can see the deep fuchsia nuclei of the chondrocyte sitting in its lacunae. And again, another real color contrast there between, the fibers outside versus the chondrocyte in its lacunae. But generally in this light blue, wavy fibers, kind of a color to it. So each one is very distinct. especially in lab, I'll often suggest, setting up a couple microscopes and have one hyaline cartilage, one elastic cartilage, one fibrocartilage, and then you have like a compact bone and a spongy bone, when we get to it. And then you just keep going back and forth, to look and compare and contrast each one. They're very, very different from one another. You just have to, practice it for about 5 minutes, not even, and you'll get it down. Okay, so now we're going to move on to bone, compact bone and spongy bone. Compact bone, when it comes to the matrix, that material other than the cells is the hard part of bone that you think of. And in compact bone, that matrix is laid down in rings, and one whole set of rings, kind of like a tree with the rings of a tree, is called an osteon. Right? So that repeating tree and all its rings is called an osteon, and it's the repeating structural unit of compact bone. And we say that because when you look at these slides, they might not be perfectly round like a tree and rings of a tree, but you will see the nature of the rings and then the big hole in the center, the central canal, and it repeats all over. And this is the beginning of looking at the composition of different organs within organ systems that have these repeating units within them because they're all contributing to the same function. Okay, so the osteons are the repeating structural unit of compact bone. Ding, ding, ding. All right. We find collagen fibers coursing either between layer and layer of lamella, which are the rings of the matrix. The collagen fibers will either travel clockwise altogether, and then the next layer they'll go in the opposite direction, counterclockwise, and then the next layer clockwise again, and they'll alternate because those collagen fibers actually allow over compression or extra compression, so that compact bone is not so brittle. I mean, when it's it gives the bone a certain amount of flexibility and withstands torsion and twisting to a certain extent. Now, children's bones have a lot of collagen fibers. So for little kids to break a bone, the stress placed on that bone had to have been quite extraordinary because they have a lot of collagen fibers. And their bones, really are kind of like Gumbies, they're kind of like rubber, and you can bend them a lot, not a lot too, but way more than adult bones for sure, because they are far more, have abundant collagen fibers within the compact bone. Okay, location is the outer rim of compact bone. So when we look at flat bones and when we look at long bones, you will see certain regions where you have compact bone. The spongy bone may be found in between that or, deep to it, certain areas, but it's generally an outer rim around all these long bones into the outer inner surface of flat bones. All right, so let's talk about our cells. I mentioned this previously. We said we have osteocytes, osteoblasts, osteoclasts. And so we will be able to predict where they're located as well. Osteocytes are found in compact bone territory, where you have these osteons. They maintain the bone matrix, located in their own little lacunae, like chondrocytes that have their own little room. Bone cells are going to have their own room, osteocytes will. So they're found in lacunae of compact bone and in spongy bone of the trabeculate. So when it comes to spongy bone, we're not going to have the matrix laid down in rings that look like a ring of a tree. They're going to kind of look like chicken wire. They're these extensions of the harder parts of the bone are going to be traveling in different directions. Osteoblasts build up bone, so B to B. They're found at the outer margins of the compact bone, where we have these two membranes called the periosteum or the endosteum. The periosteum is the outer margin of the compact bone, and the endo, meaning within, endosteum, will be found to the inner margin of the compact bone. Periosteum is superficial, endosteum is deep. And so we find osteoblasts at both of those places. And we also find osteoclasts at both of those locations. The osteoclast breakdown bone found at the periosteum and the endosteum. Now, I will tell you honestly, is that especially as you get older, they're found in higher, and especially during development, you have a higher population of or concentration of osteoblasts to those periosteums and osteoclasts to the endosteums. But as you get older, that concentration of them stops being quite as distinct. You're going to have, more like evenly dispersed osteoblasts and osteoclasts at both of those membrane areas. Okay, this is a slide of compact bone. One tree and all its rings is one osteon. Now, they're not exactly round, and if you had decreased the magnification, you would see these repeating all over. When the models will give you a better idea of that in lab. And if you actually go into the lab and look at the slides on the microscopes, it'll give you even a far more superior perspective on that. So here there's a bigger one here over to kind of the middle. And you can kind of see that there's like these rings of a tree and then a big hole in the center that's kind of blackish. And that's because there are blood vessels and nerves in there. And when this tissue dies, the blood kind of dries up and is just kind of black. So the whole tree and all its rings is one osteon. Now, each ring is called a lamella. See these little black slits? These are between lamella to lamella, between each ring. And those black slits, because there's air in there now, is a lacunae. And in life, an osteocyte would be sitting in that lacunae so it doesn't get squashed. In the center is your central canal. These are traveling vertically. This is where we find nerves, arteries, and veins. And if we could, magnify a little bit further, and you can somewhat see this on models, this will be much easier to see. But from lamella to lamella, from ring to ring, you see these lighter little grains that are traveling essentially perpendicular to the rings of the tree. And they're kind of coursing from lacunae to lacunae. Oh. Those are called the canaliculi, or can I lick your eye? And those canaliculi are narrow passageways through this compact bone, connecting the lacunae to lacunae to lacunae, essentially osteocyte to osteocyte to osteocyte, from the outer margins all the way to the central canal and back again. Why is that? Because these cells in these lacunae are alive, and they need food and oxygen just like everybody else. So their nutritional sources and oxygen sources need to diffuse from the arterial blood that's coursing in the central canal and be able to diffuse through all the canaliculi connecting to all the osteocytes in the little lacunae to essentially feed them. Further, the waste products of the activity, of these osteocytes of the lacunae need to be evacuated away. So they will diffuse from the cells to the central canal to the veins, moving down their concentration gradients so that waste material will be evacuated. Okay, so the chemical composition of bone, you have organic portions and kind of inorganic portions, but the organic portions include the cells and the osteoids. So the cells are the osteoblasts, osteocytes, osteoclasts, talked about those. And the osteoid is essentially all that matrix. It's the organic bone matrix secreted by osteoblasts that build up bone. It's composed of ground substance, which is mostly proteoglycans and glycoproteins. Right? So proteins and carbohydrate chains and proteins. Lots of collagen fibers, remember, because it's going to give that bone some flexibility and allow, some compression. Then we have the inorganic portions. So remember, organic is that it has to have carbon and hydrogen. Inorganic means it might just have one, the other oxygen or whatever. We have hydroxyapatite, which is essentially our mineral salts. Now, they are composed of calcium phosphate crystals. And those calcium phosphate crystals are responsible for the hardness and the resistance of bone to compression. It gives it the hard part of it. Hydroxyapatite is about 65% of the bone mass. just for those who want to know. Right. So the hard part, the hard part of the bone is going to be the hydroxyapatite, and it's mainly composed of calcium phosphate crystals. Okay, so that is our compact bone. All right, let me just go back there again. So we have osteons, that is repeating structural unit of compact bone, not spongy bone. The rings here of matrix are called lamella. We have lacunae, the dark slit areas there. That's where we would find an osteocyte. The little, looks like it took a pencil and drew little lines perpendicular to those lamella connecting lacunae to lacunae to lacunae to the central canal. Those are the canaliculi, or canaliculi, and the central canal is where we find nerves, arteries, and veins supplying this bone. So we're going to be, in lab, we see a lot of those pics there. All right, so we're going to go to spongy bone. Now, spongy bone is found elsewhere. Spongy bone is found in flat bones. It is found, it's called the epiphyses of long bones. So long bones are very abundant in our body. And we're going to look at that because, you have regions of the bone. The middle area is called the shaft or the diaphysis, and the ends of bone, which are the knobby parts that essentially connect one bone to another. Those are your joints or articulations of the body. Those are what's called the epiphyses. And so it's common to find this spongy bone throughout the epiphyses, proximal and distal, the two ends of the long bone. We find a lot of spongy bone there. But we can find the spongy bone called diplo between within the middle of flat bones as well. Okay, so spongy bone has two major components to it. We have the trabeculae and the red bone marrow. The trabeculae are the finger-like projections of the bone, the hard part, the more resistant matrix that we looked at in rings in compact bone. Well, in spongy bone, it's going to look like lattice fencing or a honeycomb kind of an appearance. And so the walls of that fence or the honeycomb is going to be the trabeculae. And it's in there that you will find some of those osteocytes maintaining that matrix. Now, because they might be narrow in diameter, they are very abundant and coarse in different directions. And because of that, they help to resist a lot of stress in those areas. It also helps to translate forces from, like a lateral aspect to a medial aspect. Is going to help with that. It's lighter than compact bone, so it helps reduce the weight of the bone. Right, found between the trabeculae, right, it's found between the walls of, the honeycomb or, the lattice fencing. We're going to find this red gooey material or this granular material, and that's your red bone marrow. Right? Red bone marrow is where all blood cells originate. Red blood cells, white blood cells, platelets, they all originate from red bone marrow. All right, so spongy bone is always composed of those two things, always. Okay, so if here we have a picture, and in the upper left-hand corner, they're showing a little square from one of the parietal bones. That's one of your flat bones of the skull. And when we magnify it, you go, oh, look, here, just below that, it kind of looks like a bone sandwich. You have a thick, and it looks, completely dense. Compact bone to the undersurface and the upper surface. And in between, sandwiched between those, is the spongy bone. And in these flat bones, we've actually referred to it as diplo. So this is dehydrated. There isn't any red bone marrow, but you see now the honeycomb nature that I was talking about. In life, you would have red bone marrow stuffed in there, but this is like a model or a bone that used to be full of life, and now all the watery parts have dehydrated, and you're left being able to see the walls of the trabeculae. So in that is all the trabeculae. Below that, it magnifies it even more. So it says trabecular spongy bone. You see how it's coursing in different directions? Okay. In life, you would find, fat tissue in there, but that's where you would find red bone marrow. Over to the right-hand side, right, it's severely magnified, one of those areas that still has red bone marrow in it. So the pinky, the pink kind of Y looking structure in the center there, looks kind of like a Y. That's a trabeculae. And the black staining nuclei, those are some osteocytes in their matrix. Right? So instead of laying the hard part in rings like a tree in compact bone, this is going to be like a lattice fence, kind of honeycomb orientation to the hard parts. Found between them is going to be, you see the dark purple granular material? That is the red bone marrow. And the other pale staining big cells there are fat cells, adipocytes. It's very common to find that, in between the trabeculae of spongy bone. So the two components of the spongy bone are the light pink Y, that's the trabeculae, and the purpley greeny material of red bone marrow. Okay, so when it comes to bones, we have shapes. We have long bones, short bones, flat bones, irregular bones. And we often look at these in lab as well. Long bones are longer than they are wide. They have a shaft and two ends called epiphyses. They're common throughout the body. And this includes all the limb bones. Short bones, or cube-like bones, mostly composed of spongy bone. The bones in the wrists and the ankles, the carpal bones and the tarsal bones are great examples. And your kneecap, the sesamoid bones in your body, and the largest sesamoid bone is your patella or your kneecap. So those are great examples of short bones, because short bones are different from irregular bones. They look similar, but they're not technically similar. They're not the same. Right, flat bones, bones of the skull. We just looked at one. Bones of the skull, those are going to be flat bones. Your sternum or your breastbone, ribs are flat bones. Parts of your scapula, they're considered flat bones. So they're thin, flattened with a slight curve, possibly that resembles a sandwich of compact bone with spongy bone in the center. Okay, just like we looked at right here. All right, then we have irregular bones. These are complicated shapes that are mostly spongy bone. The vertebral column, all the vertebrae of your spine, they are all individually shaped. They follow general similarities from region to region, like cervical vertebra, thoracic vertebra, and lumbar vertebra, but they're actually all unique. And you have three bones on each side of your pelvis. These bones, an ilium, an ischium, and a pubis bone, come together and fuse and form each side of your pelvis, called an os coxa. And those are irregularly shaped. Right? So a vertebra, the vertebral column, is an excellent example. All right, so here in this picture, it gives you examples. A long bone, this is the humerus, and it shows you it's coming from the upper arm, but the radius and ulna of the lower arm, they're long bones. We have the femur, the tibia, the fibula, other examples of long bones. That's A. B is the irregular bone. Again, the vertebral stack, excellent location to keep in mind. Flat bones, your sternum, bones of the skull up here. You can't see the scapula, but that would be to the backside. And short bones would be bones like from the foot, really composing more of the ankle area, the tarsal bones and the wrist bones, which are called the carpal bones. You walk on tar with your tarsals. You drive a car with your carpals. That's how you're going to remember that. Okay, so we're going to look at these long bones because these occur very frequently throughout the body. And I will tell you that long bones, there are some long bones, like in your fingers, your phalanges, the bones that make up your fingers and your toes. They might be small, but they are a long bone. I will just tell you that's one of those kind of freaky things. Long bone doesn't necessarily mean it's the biggest bone. It's how it develops embryologically, and whether, again, and then therefore it would have, even if it's tiny, like one of the little bones of your toe, it would have two epiphyses and a diaphysis. Okay, so what is the diaphysis? That's the shaft of a long bone, the long axis made of a thick collar of compact bone surrounding it, and a medullary cavity to the center. So I'll show you what that looks like. That cavity will either be filled with red bone marrow or yellow bone marrow, depending on the age of the individual. The ends of that long bone are called the epiphyses. They have compact bone to the outside and spongy bone to the inside. To the outer surface of that epiphysis is going to be cartilage, hyaline cartilage, that helps cushion the bone ends during joint movement. And between the epiphysis and diaphysis, depending on the age of the individual, you will either have an epiphyseal line or an epiphyseal plate. Right. So what does that mean? There is an area between those two, kind of called a metaphysis, where when you're young and your bones are still growing in length, we have an epiphyseal plate there. And that means that we are going to have hyaline cartilage there that allows these bones to lengthen at both ends, lengthen the bone a little more rapidly that way, or a lot more rapidly that way, during childhood. Once you reach skeletal maturity as an adult, that cartilage is going to be replaced by bone, and it then becomes, or it's called an epiphyseal line. So an epiphyseal plate is an open growth center. I guess you can, that's a term you might be able to identify with, because we have hyaline cartilage there, and that's our area of bone elongation. But once you mature, that cartilage is going to be replaced by bone, and it's called an epiphyseal line. That's why when you get to a certain area, you stop growing. Your bones cannot lengthen anymore. You know, it starts to be remodeled. Your hormones are going to change and so forth. And so once that's replaced with bone, it will not elongate again. Okay, so here we have a picture over to the left-hand side showing you region. It's a long bone. You have an epiphysis called the proximal epiphysis. And this is just depending on what end of the bone or the other, you know, as far as anatomical position where it's going to be found. So there's always going to be one proximal epiphysis, the diaphysis, and a distal epiphysis. Right? All right, now, so let's look at that. The proximal and distal epiphysis, they cut into the proximal one here. You have, first of all, you have light blue at the ends, called the articular cartilage, because that is hyaline cartilage. And then you have spongy bone deep to the compact bone rim. So all the way around this long bone, whether you're in the epiphysis, the diaphysis, or the distal epiphysis, right, is that the outer margin there looks completely solid because that's compact bone. Now, you know, we're going to look at models of compact bone and realize bone is not solid even there. There's actually microscopic tunneling systems through it all, but macroscopically like that, it looks solid, you know. All right, so that surrounds the entire margin, the outer surface of one of these long bones. When you're at the epiphysis and you go deep to it, look at all that trabeculi. All right, so that is the spongy bone. The hard part of the bone you're looking at there is the trabeculae. And, you know, if there was red bone marrow in there, you would see that with like this grainy appearance of dew in there and some fat. Okay. Further down, so and then she actually takes a piece of that and has an arrow over to the right for the upper right hand corner there, and that represents the trabeculae, you know, from that region, the compact bone region to the outer margin, and then the light blue hyaline cartilage making up the articular cartilage. All right, we'll come back to the endosteum, periosteum in just a second. Okay, so that's what that arrow is trying to show you. Now let's look at the diaphysis. If we open that up, we see this cavity to the interior. The margins to the right and left are your compact bone, looks like solid. In the center, you know, there's a cavity there. Well, when you're young and you're just a child, an infant, you know, in child growing until a certain age, you have red bone marrow in there too. But as you get to skeletal maturity, and you know, at that point, your volume of blood isn't increasing dramatically. You just have to maintain kind of the same quantity of blood. Kind of a long story. We talk about that more at A&P2. Is that we will replace the red bone marrow there with yellow bone marrow, which you can see down below. And so, what does yellow bone marrow do? Well, yellow, you know, fat areas are often indicated in yellow. So just think fat from the chemistry lab, long-term energy storage, cushion and insulate organs. Okay, you can see the compact bone to both sides. And then you can also see periodically these arteries piercing through there. So this is kind of good to keep in mind. We have what's called these nutrient arteries that are going to enter into, you know, because they have to start from the outside of the bone and eventually get to the inside of the bone. Is that this is how we serve, you know, we provide a blood supply to all this compact bone and through some of the spongy bone there. Arteries are going to enter in there, and then it's going to branch off. And you're going to have as many branches as you are osteons with a big central canal, right? And also to the outside margins. So it comes in as one big artery, but then it's going to branch off into these very small arteries, supplying all the compact bone, all the osteocytes, osteoblasts, and osteoclasts, essentially. Over to the lower right-hand side to get a little better idea of what that would look like. We have these openings there for the arteries to pass through. To the outer margin of the compact bone, you see the whitish kind of lacy material to the outside, right? That's the periosteum. We said that there's a membrane to the outer margin of the compact bone and the inner margin. So the one to the outer and around is called the periosteum, where we will find osteoblasts and osteoclasts. In compact bone, in all those osteons and the little punes, that's osteocyte territory. Right? Then to the inner margin, before we get to the yellow bone marrow, is an endosteum, where we find again more osteoblasts and osteoclasts. Let's see, what else was I going to say about these? I think I think that's okay. That's pretty good. Okay, so here are the membranes. The periosteum, double-layered membrane covering the diaphysis to the outer margin of the compact bone. It provides anchoring, and it has a couple of functions. So it's not only where between that and the compact bone we're going to find the osteoblasts and osteoclasts, but this is like the connective tissue that surrounds the particular organ, which is going to be a long bone. And we keep saying that connective tissue surrounding organs always pretty much serves the same function all the time. It helps provide an attachment point for other things. So those arteries and veins entering and exiting, and the nerves entering, and exiting the bone need to be anchored on its surface. So this connective tissue, this periosteum, helps do that so they don't get yanked away. It's also a good surface for the tendons of muscles, because tendons attach muscle to bone. This is a good surface for that tendon to attach to, one connective tissue layer to another connective tissue layer. All right, so it provides anchoring for tendons and ligaments that attach bone to bone, nerves and blood vessels. This will really contain the osteoblasts and osteoclasts for bone remodeling and growth in diameter. So the osteoblasts and osteoclasts work in opposition to each other. One builds it up, one breaks it down. So it depends on what phase of your life you're in. Am I still growing? Are you just a child or an adolescent? Did I fracture a bone as an adult? Am I just perimenopausal, postmenopausal? Or for men, by the time you get into, your later 60s and 70s, your bone's demineralizing as well, right? So, it depends on what stage of life you're in, which cells are going to be more active, and maybe what stage of, let's say, a bone fracture repair you're in. All right, so periosteum, outer layer of compact bone. Endosteum is the internal the membrane. Lines the medial layer of the compact bone, or the deep layer of the compact bone. And then on the other side would be the medullary cavity. It too contains osteoblasts and osteoclasts for bone remodeling. So both cells are found in both areas. Okay, again, short, irregular, flat bone gross anatomy. Contains bone marrow, but no medullary cavity. So again, it was that bone sandwich. You had compact bone to the outside, to the inside, and then spongy bone found in between, or the diplo found in between, like we saw in that parietal bone. Okay, hematopoiesis is blood formation. We said that all blood cells come from red bone marrow. So that is called hematopoiesis, right? Found in red bone marrow. In children, red bone marrow, is found in the medullary cavity of long bones, in the humerus, in the sternum, and in the areas where we're going to find it in adults too, at the epiphyses, at the ends of these long bones. In adults, those medullary cavities are going to replace the red bone marrow with yellow bone marrow. So you might get a question pointing to, a medullary cavity on a model, and the question might say, identify this structure, and you'd say, medullary cavity. And so maybe the question would be, what material would I find here as an adult? Yellow bone marrow. What material might I find here in a child or an infant? Red bone marrow. Now the epiphyses, now we're talking about medullary cavities. Don't get confused. When it comes to spongy bone at the epiphyses like that, is only going to have two major components. The trabeculae, which are the walls of the, that you can see of the spongy bone, and the grainy goo found between it all, the red bone marrow. That will never change. All right. A spongy bone is composed of trabeculae and red bone marrow forever and ever, wherever spongy bone is found. But the medullary cavities of long bones in the diaphysis may be different depending on the age of the individual. Okay, so we're going to come back to this now, osteon of compact bone, because now we can look at some models of it. All right, so again, the osteon is the repeating structural unit of compact bone. It looks like a tree and the rings of the tree. So those rings are called lamella. This is how the matrix, the hard part in compact bone, is going to be arranged in rings like this, with a big hole in the center called the central canal that contains arteries and veins and nerves. The lacunae are the little cracks and openings between the rings of lamella, that will house an osteocyte. The osteocytes maintain the bone matrix there. And so they're found in compact bone territory. And the canaliculi are the little narrow diameter passageways, essentially connecting each osteocyte to each other and ultimately to the central canal, to the blood supply, where nutrients can go from the blood out to all those cells, and the waste products of the cells can go from the cells to the central canal and be absorbed in the veins and evacuated. Okay, so here we have this long bone. It's a femur. So over the left, upper left, to show you essentially, I'm going to take this little wedge with the gray arrow and magnify that. All right, so it's taking part of that compact bone region into the medullary cavity. All right, so they're going to magnify that. So the outer margin, I kind of start from the outside, in gray is your periosteum. And you can see some nerves, arteries, and veins passing through it. The red, the blue, and the yellow. Okay, then you go deep to that, and you now see the hard matrix being laid down. You can see these repeating osteons, right? Rings of a tree. In the center of each, you can see a red, blue, and a yellow nerve, artery, and a vein. Over to the right-hand side, it cuts open a couple of those osteons, so you can see how the blood vessels and nerves are coursing. Right, so these arteries and veins are going to pass through what's called like a perforating canal or a Volkmann's canal. They're going to go from the outside of the bone into the bone, and they said then it's going to branch off like a highway system, so that in the center of each osteon, through each central canal of every osteon there, is going to have its own vascular supply and a nerve supply. And I think it's really cool to see that, because they're going to branch off in different directions, and every single one is going to have an arterial, vein, and nerve supply in the center of every central canal of every osteon. Okay. Then to the medial aspect, of course, I should say the deeper aspect or medial aspect of the compact bone region will be your end osteon. All right, so it has that more in like the darker kind of brownish region. It has endosteum lining the bony canals and covering some trabeculae. Okay, here's a slide. Here's one really magnified here on the left side, and the right side shows you what a typical osteon of compact bone would look like. You can see the rings called lamella, central canal, nerves, arteries, veins. You can see the lacunae, which would house an osteocyte in life. You can see the little passageways. It's kind of like taking a pencil and drawing lines radiating from the central canal outward, like spokes on a tree, spokes on a wheel, on a tree, spokes on a wheel, a bicycle wheel. And that's the little passageways that allow material from each lacunae and osteocyte to the central canal and back. Okay, so then we're going to talk about osteogenesis. Right, so this is the formation of the bony skeleton. So you have two major methods, intramembranous ossification and endochondral ossification. And then we have a method of increasing bone length as well as diameter. And when we increase the diameter of long bones, especially, it's called appositional growth. We look at the development more of long bones and flat bones. Okay, intramembranous ossification, endochondral ossification. All right, so intramembranous ossification is the method in which we form flat bones, like the skull bones, a lot of the bones of the skull, your clavicles, your scapulas. Right? Now, they develop differently. So flat bones develop from fibrous connective tissue. Fibrous connective tissue usually begins in the center of the membrane called the ossification center. And that's all you have to know about it, because it's not really that dramatic, but long bone development, is a little bit different, or actually quite a bit different. So intramembranous ossification is the embryological, while you're silly putty in your mother's uterus, how your flat bones are going to develop, right? And they develop from a base of fibrous connective tissue. Right? This is how our long bones will really develop, and they do that through endochondral ossification. We develop from a hyaline cartilage model. What does that mean? It means that you're going to have a mass of hyaline cartilage, chondrocytes in your lacunae, and it's going to be, I don't want to say a blob. To me, when in the pictures, they kind of look like a great big milk bone for your dog. You're going to have this mass, and as it gets bigger, all right, and that mass goes from small to larger, because we're all at that age growing, even embryologically. You're going from a very, very tiny, tiny little dude to a larger dude before you're born, is that they're going to go through this process. Right. But we start with hyaline cartilage. So it's going to be in the light blue color. Right. It develops ossification centers, in the diaphysis. We have a primary ossification center and a secondary ossification center. So I'll show you what that's going to look like, because the primary ossification center is the area that changes from cartilage to bone and stuff first. And then the secondary ossification centers goes through a similar change from cartilage to bone, but this is going to occur at the epiphyses. At the end of ossification, hyaline cartilage remains at certain areas. And these are the areas we use to be examples of hyaline cartilage locations, at the articular ends of the bone. So we saw that on one of the first long bone pictures. So look, you've got the blue at the two ends. It helps, keep the bones, from eroding when they articulate with another bone over years and years. And at the epiphyseal plates, where the diaphysis and the epiphysis meet, where we have bone elongation. And we said, as long as there's cartilage there, we call it an epiphyseal plate. When you reach adulthood, that cartilage is replaced by bone, so we call it an epiphyseal line. Okay, so there's actually a stage over to the left preceding this that the book used to include, and they don't include it anymore. But it would be one solid mass of light blue soap bubbly stuff there, and that would be hyaline cartilage. All right, one goes to the next stage. At a certain point, as this blue, mass of hyaline cartilage gets larger and larger, and just by the way, before we even go any farther, the difference between the size of image 1, 2, 3, 4. This is bones getting larger. All right, so you have to picture that this is actually getting larger over time. Okay, so what happens is as this, what was just the stage prior to this, entirely light blue, soap bubbly hyaline cartilage, it begins to now develop what's called this bony collar. So to the waist part here, like of an hourglass, you start to develop bone there. And when that happens, the chondrocytes in the lacunae, in the central region there, see, throughout the hyaline cartilage here, those chondrocytes in those lacunae, they need food, just like any of those osteocytes did before. but they get their oxygen and nutrients and things by diffusion from the fluids that are found outside of it. Right? So, as this model and this mass of cartilage gets larger and larger, you have to picture the fact that to feed the cells in the center, you have this longer diffusional pathway. When we're talking about things that accelerate diffusion and enhance it versus inhibit diffusion, in the previous chapter, 2 chapters ago, is that we said the longer the distance you have to diffuse, the more it slows it down. And actually, as you pass through more and more cells, they essentially take all the oxygen and take all the food, so that the cells in the center here, that are being cut off by the, the bone here and so forth, essentially start to die off because they don't have any nutrition. I mean, that is the first area where we go from hyaline cartilage to bone. Right? So we call it the primary ossification center. So number one, bone collar forms around the diaphysis of the hyaline cartilage model. Now remember, the stage prior to this, if there was like a one before one over the left-hand side, you just have all like lip. And they're pretty much able to sustain life that way. But this bony collar starts to develop, and it cuts off the food supply, and it starts to die off, all right? So it's going to develop a cavity. That takes us to number 2. This is the beginning of a medullary cavity. Right, so cartilage in the center of the diaphysis calcifies and then develops cavities. So what does that mean? It means the chondrocytes die and they get replaced by osteocytes. Because if you're going to be replacing with bone, even spongy bone, you have to have some osteocytes. Right? Then it gets larger and more pronounced, and it encourages a blood vessel to bore through the bony collar and branch off. So this is the beginning of the branching system to supply the center of all those osteons. Okay, it gets larger, clearly, and larger until finally we get to number 4. The same sort of phenomena that happened in the center of the bony collar is going to happen at the 2 ends, which become the epiphyses. So this area of cartilage gets thicker and larger till, again, the diffusional pathway from nutrients outside the blue area to all the cells in the center, it just is too vast. And all the other surrounding cells essentially eat everything up, right, for all intents and purposes. So the interior of the ends go through that same process. The cartilage cells die off and they get replaced by bone cells. That is followed by blood vessels boring through this, the hyaline cartilage here and branching off to supply all the osteocytes there. And it does it at both ends. It gets bigger and bigger until finally, and it says on the top, from childhood to adolescence. This is a classic long bone. You have your 2 ends called the epiphysis, the shaft in the center of the diaphysis. You have a blood supply to each end. It doesn't show it at the distal, epiphysis, but it shows it here in the medullary cavity. And we're left with our 2 remaining hyaline cartilage light blue areas, the articular ends of bone and the epiphyseal plate. When you reach adulthood, that epiphyseal plate here will turn white because it's those cartilage cells die off, it gets replaced by bone, and it becomes an epiphyseal line. Okay, so here in number 4, the diaphysis elongates and a medullary cavity forms. Told you, secondary ossification centers start to appear in the epiphyses. The epiphyses ossify, which means it goes from chondrocytes to osteocytes. When completed, hyaline cartilage remains at those 2 locations. And this starts here. Look at the headings in black at the top. Week 9 of embryological development. Month 3, birth, childhood to adolescence. And so this process continues well on into life. Okay, the growth in length of bones. So let me just go back here. All right, so you're born with this little bone here. It says here is birth. Right? That sucker's got to get longer and wider. The bones you're born with are very narrow in diameter and very short. Thus, that's why infants are very tiny. But all those bones need to lengthen and widen so they can be more weight bearing. So those 2 phenomena have to happen. So this occurs at those epiphyseal plates. We said the growth plates. From those two ends, here we'll go back from the proximal kind of epiphyseal plate and the distal epiphyseal plate. This is where we're going to lengthen the bones dramatically, all right, so that we somewhat increase the size of the epiphyses, but we certainly increase the length of the diaphyses. And then we're always going to be remodeling a little bit so we don't end up with these great big bubbles at the end, that the epiphyses stay, proportional to the diaphysis here, and that the diaphysis really lengthening, and that we also then have to widen it. Okay, so that's what we're talking about, lengthening at both of those ends. Okay, so this occurs at the epiphyseal plate. We have a region, that part of it that's hyaline cartilage, that's called the zone of resting cartilage, which is the majority of what you're visually seeing there. Right? Between that cartilage area and like the point where it meets the diaphysis, then our next zone is going to be our zone of proliferating cartilage. So here, we're going to, instead of in resting cartilage, it's going to have that soap bubbly appearance of hyaline cartilage, chondrocytes and the lacunae. The matrix is light purple, maybe light pink staining with no visible fibers. But when you get to the proliferating cartilage, as we go behind that, we are going to see an area of the cartilage where these chondrocytes are rapidly increasing in number. And that's called proliferation, increase in number. And they're going to stack upon themselves. Instead of being dispersed, kind of like soap bubbles all over, they're going to stack on each other, 2, 6 at a time. We call the stack coin appearance. Behind that and traveling toward the medullary cavity, to the diaphysis, those proliferating cells will then morph and start to die off into the hypertrophic zone, so our zone of hypertrophy. And what's happening here is those chondrocytes are starting to die off because they're going to be replaced by bone cells. And as they do, they start to increase in size. They start to have what I call the chubba-lubba cells. They have a lot of cytoplasm, within the cell, and they have a very translucent appearance. That then is followed by calcified matrix. Right? And this is where now we have transitioned from cartilage to bone cells. And this will be a beautiful example of spongy bone. So this is where cartilage cells die and are replaced by spongy bone, by the osteocytes that are now making spongy bone. This occurs primarily during adolescence until skeletal maturity is reached. The epiphyseal plate closure, fusion of the diaphysis and the epiphysis, occurs, generally around 18 years of age in most females. This is the average. 21 in males, actually. So by the time you're in college or running around and playing sports all this time, some of you guys, your growth plates are still open. You are going to be susceptible to certain types of fractures, actually, epiphyseal fractures and things like that. And for women, the same way. If they're playing more intense sports early, like in high school, they can suffer from the same issues. That epiphyseal plate becomes an epiphyseal line or a closed growth plate. Okay, so here we're going to see these zones, and I used to like this picture better because they showed you what the cartilage, the normal looking hyaline cartilage above it would look like. They call it the resting zone above, and they kind of, so we'll start with resting zone. You're going to picture up here that soap bubbly appearance of hyaline cartilage. And on the upper left-hand corner is showing you where this is coming from. All right. So to the top region would be your light blue, beautiful, hyaline cartilage, so bubbly appearance. Below that, those cells, all right, as the ends of these bones advance forward, all right, or away from the center, let's put it that way, away from the medullary cavity, right, those cartilage cells start to proliferate. So behind that, you're going to have this zone of proliferation where the cells stack up on one another. That will be followed by a hypertrophic zone or zone of hypertrophy. You see now, up on the top there, it almost looks like quarters or nickels stacked on one another. But you get to hypertrophy, and look, you can see these great big cells, kind of in their lacunae, very large and translucent. I call them the chubba-lubba cells. right? Those are dying off and then they get replaced by, oh, our zone of calcification and then further ossification. So we kind of link those together, right? So where these cartilage cells die off and they get replaced by osteocytes in the trabeculae and spongy bone. And so now kind of like look down in there. It's light pink walls of trabeculae, and then you have this grainy goo, so to speak, and dark purple nuclei there. That would be the red bone marrow. If you could go a little bit farther, below where it says for ossification, a little farther down, about another, millimeter or two, then that tissue there would look like beautiful example of well-established spongy bone. Okay, so those are those 4 zones. We have the zone of resting cartilage, zone of proliferation, hypertrophic zone, or zone of hypertrophy, and calcified matrix. Use that sequence of 1, 2, 3, 4. I know in this image it says resting cartilage above, and then it has proliferation as number 1 or 2. Just disregard that. We use resting cartilage, resting zone as number 1, proliferation number 2, hypertrophy number 3, and we link together calcification, ossification, because it's essentially, this not the same exact thing, but it's happening very, very close to one another. Okay, that's how we lengthen the bones. Now we want to increase the diameter of bone, and that's called appositional growth. This requires the coordinated activities of osteoblasts and osteoclasts. If I want to increase, the width of the bone, increase not only the epiphyses, because those have to get larger, but especially the diaphysis. Right? And we don't want to just lay down a whole bunch of bone on the outer surface, because then your compact bone region will be really thick and heavy, right? So you want to lay down a lot of bone at that periosteum using the osteoblasts. And then to the inner margin where we have the endosteum, you're going to let the osteoclast break down a lot of what you just built up. All right, so that the thickness of the compact bone that surrounds the entire long bone, it's going to get thicker, because clearly it's going to have to be thicker than bone, like a femur of a 1-year-old is going to have to be thicker than an of an adult. But you don't want to just do that. So, yes, the compact bone rim is going to get thicker, but what's really going to get larger is the medullary cavity. And as you get older, especially before, while you're still a child, you want that area to expand. You want more red bone marrow there, because you need to make a lot of bone cells, for the bones that are dying off every 120 days, which is, the red blood cells, which is normal. But you're trying to increase the overall volume of blood from maybe 1 liter to 1.25 liters to 1.5 liters to 2 liters as you get larger and larger and larger to about 5 liters as an adult, and you have to constantly replace those. You're increasing the overall volume as well as replacing that which dies off about every 120 days. The growth of bone is hormonally regulated by, minimally these 3 hormones: growth hormone, thyroid hormone, and sex hormones. And I will say that it is heavily influenced by sex hormones, estrogen and progesterones in females, and testosterone in males. Growth hormone from the anterior pituitary, and we'll look at growth and thyroid hormone and testosterone, and estrogen, progesterone more when we get into AMP2. But growth hormone helps stimulate protein synthesis and recovery of tissue that's get used, all day, every day. So it helps cells like really recover. This is really good hormone. It's released during REM cycles of sleep. And this is the kind of hormone you really want to have a lot of, maybe napping frequently when you're a really serious athlete, because you're kind of training really hard and kind of breaking down tissue, so to speak, and you want to repair very quickly. So you want to be releasing a lot of growth hormone. Thyroid hormone helps regulate growth hormone for proportional growth. So what do I mean by that? T3, T4 are 2, arguably, different hormones that are made in the thyroid gland. And then there's another one called calcitonin. But thyroid hormones, T3, T4, stimulate metabolism. It regulates the rate of which cells can convert different substrates like glucose and stuff into ATP, and to have plenty of pumps and to recover. So the rate in which we metabolize effectively and burn substrates, is going to be a lot of thyroid hormones. So these all work together, and especially with sex hormones. When those hormones start to elevate, in early childhood and adolescence, that's when you start to see kids go through their growth spurts. They're developing secondary sexual characteristics. They're getting all hormonal and crazy. Right, so that happens all at once. So sex hormones have a big influence on bone elongation and things, but so does growth hormone and thyroid hormone. Okay, bone homeostasis. This is the basic processes of remodeling. Osteoblasts and osteoclasts work together, but in opposite effects. We have bone deposition, which is how you lay down new bone, and that's the function of an osteoblast. Build up and lay down bone. Occurs when bone is injured or added in strength, or, when we're trying to increase appositional growth, when we're trying to increase that. But, we're sort of talking about also, here is that, okay, well, what happens after you reach skeletal maturity and you're an adult though? Well, we do have different opportunities to bone deposit or bone resorb. If we're going to bone deposit, which we may have to do because of you're repairing a fractured bone, then the osteoblast will be stimulated. If we want to break down bone, then we want to stimulate our osteoclasts because they resorb and break down bone. They secrete lysosomal enzymes. So osteoclasts have a lot of lysosomes, and they help break down the bone, the hard part of bone, the mineralized bone, so that the calcium and phosphorus and things that are the minerals stored there can enter into the bloodstream. So there's different phases of bone growth where you're going to be laying down bone at some point and sometimes breaking it down. When we're increasing the diameter, like we were saying before, of a long bone, then we stimulate the osteoblasts on the periosteum and the osteoclasts on the endosteum, so that we don't have one great big super thick femur. We have a big medullary cavity in the center. But we can also, throughout life, stimulate or inhibit these cells just to regulate the amount of calcium in our blood. Calcium is used for muscle contraction, of skeletal muscle, cardiac muscle, smooth muscle. I mean, you're going to need calcium all over. All right. So, and, it wears down, it becomes kind of inert. So just because you started with a certain amount of calcium doesn't mean you're going to have it later. All right. But we can store excess in compact bone. So that's why, there's a lot of calcium in milk, right? And one of the commercials for milk used to be, build strong bones and bodies. Well, it actually does, because you need to have calcium-rich foods, when you're growing in adolescence, and then to maintain proper, composition of bone in adult life, because we use calcium for other things as well. Right? So those other organs can take the calcium, and then we need more. Well, if we didn't eat enough in our diet, we need to go back to the bone, and break it down with osteoclasts so that we can take its calcium. And we'll talk about, some of the problems with that, so to speak. Right. Well, that takes us to remodeling control, because this part here, and this gets hit on, you'll get this again in the endocrine system, and so forth, is that we will talk about this, about blood calcium regulation. Right? Because we need calcium for other things other than just maintaining good density of bone. All right, so we have varying levels of calcium. When blood calcium levels drop lower than normal, right? And that's why, in nursing, you have to know normal ranges of things, because some things have larger fluctuations of normal. Some things are very narrow, like blood pH is 7.35 to 7.45. Not a lot of wiggle room, right? But sodium and chloride, and so, you have much larger highs and lows to the range, right? But when you go outside of the ranges, it means you need to do something as best as possible. Some are very, very sensitive to changes, and then some, it's not so dramatic if it drops low or high, but you will try to, again, restore it. All right, but calcium is one of those. Blood glucose levels is another one. We look at that, pretty significantly in AMP2. All right, so let's say your blood calcium levels start to fall lower than normal. The parathyroid gland, which is found on the back of your thyroid gland, which is kind of in your throat area there, secretes a hormone called parathyroid hormone. Shocker, right? So parathyroid hormone, or PTH, is released because calcium levels are low. So this is what you want to do. You want to raise calcium levels from different sources so it comes back up into a normal range. And then you stop secreting the parathyroid hormone because, like most things, it's a negative feedback loop. All right. So what does that hormone do? It stimulates the osteoclast to release those lysosomal enzymes and resorb bone so that the minerals there, like calcium, enter into the bloodstream and you start to elevate blood calcium levels. Oh, but that's not all. It also targets the kidneys to reabsorb calcium that's passing through the kidneys where you're forming urine. And it also, at the kidneys, converts that vitamin D that we talked about in the integumentary system to calcitriol. And we say calcitriol targets the small intestine to absorb calcium. So what ultimately happens? We elevate blood calcium levels, right? So we start with low blood calcium levels. We use parathyroid hormone via these ways, right? And we ultimately end with elevated blood calcium levels. Now, the opposite can happen. We can start with, higher than normal blood calcium levels. Well, now what are you going to do? Well, that thyroid gland can release calcitonin. And calcitonin inhibits osteoclasts and now stimulates the osteoblasts to deposit calcium. All right, so your calcium levels are higher than normal. You want to decrease it because, again, whether you're high or low, normal ranges are normal ranges. So if it's high, you want to get rid of some excess. And sometimes what's happening is that you have these osteoclasts going to town and, whatever, they're just not shutting off. Or you just ingested a lot of very calcium-rich foods. Okay, so in either event, if it's high, we can at least temporarily inhibit the osteoclast, kind of put a muzzle on them so they stop degrading that compact bone and releasing more calcium into an already high blood calcium level. Right. So we inhibit the osteoclast. Well, heck, now that we have an abundance of calcium, let's stimulate the osteoblasts to take that extra calcium and let's store it in that compact bone for later. So that when you have an excess of something, you want to fully pack in and store as much of that as possible for later. Right? And so by those two things, and then blood eventually circulating to the kidneys and letting the excess calcium flow out in urine, you decrease the blood calcium levels back to a lower level. And back into a normal level, really, I should say. And then you stop secreting calcitonin. All right? So you should know these negative feedback loops between calcium, high blood calcium levels and low blood calcium levels. Which hormone is used, parathyroid hormone, or calcitonin. And know their basic mechanisms. If you know it now, that's just 2 more hormones when it comes to the 12 or 13 or so you need to know in the endocrine system. You're already now 2 ahead. Okay, and this just shows you how we balance up at the top on that seesaw, calcium homeostasis. Normal levels about 9 to 11 milligrams per 100 liters. Over to the right-hand side, it says stimulus, falling blood calcium levels. So calcium levels are lower than normal. It's showing you this thyroid gland here. And from the front, it just looks like all thyroid. But if we look at the back side of it, you can see these little marbles kind of in there, and those are your parathyroid glands. So the parathyroid glands release parathyroid hormone. We just talked about this targets. It's going to target the osteoclast, the kidneys, convert vitamin D to calcitriol to absorb more calcium, and thus we increase blood calcium levels back to normal. Over here, they show you some osteoclast. Doesn't it look like a Pac-Man? I think it looks like a Pac-Man. Osteoclast degrade bone matrix and release calcium in the blood. But that's not its only, that's not the only thing the parathyroid hormone does. And so we want you to know the other mechanisms now so that you start getting it down for AMP2. But I think those look like hack mail. Okay, fracture repair, FYI. I will say honestly, I don't ask you really questions about this, but I think it's really good to look at, and see, because most people, not everybody, I've never had a fractured bone, but most people will, or someone they know is going to experience a bone fracture. So it goes through these 4 phases: hematoma formation, which is when the bone cells eventually die due to lack of nutrients. Remember, when there's a lot of blood vessels coursing through the compact bone and through the spongy bone. So when you fracture a bone, aren't you cutting off those those blood vessels? You're cutting them, right? You're going to hemorrhage to a certain extent, bleed, which forms a bruise, which is what a hematoma is, right? Accumulation of blood outside of the cardiovascular system like that, right? So you're going to essentially have a mass of blood there because the blood vessels have been cut as well, and then the bone cells that it supplies, the areas of bone, those cells are going to die off. It's followed by swelling and inflammation can occur, where eventually those blood vessels that got lacerated, so to speak, you're going to have vasospasming around there. So you'll bleed some, but eventually you start to block that all off, with clotting and things. And you get swelling and inflammation because you have pain, you have fluid accumulation, is part of your immune response to have this swelling and inflammation occur. That's followed by the granulation, or tissue granulation that forms. Fibrocartilaginous callus is followed by a bone callus. So what happens? Where these 2 bones need to approximate one another, you're going to put down a good amount of fibrocartilage there that forms this scar tissue, essentially, or a callus. And then that will be remodeled cartilage replaced by bone cells, with bone remodeling. You're going to reshape the bone, pull the edges of the bone back together again, or lay down some new bone that kind of bridges that region. If it's reset well, then there's not as much new bone to lay down. But it goes from like a cartilaginous callus to a bony callus, and then your osteoblasts and osteoclasts are going to work in there to try to build up the bone and then kind of sandpaper it, like taking a nail file to it and getting it as smooth as possible, and back to normal, its normal shape, right? So we start with a hematoma, you get bleeding in there, and then you have this fibrocartilaginous callus that forms. Then we replace that with bony callus and bone material. And then you kind of come back and let your your osteoblasts and osteoclasts are working to lay down bone and then break down the excess of bone so that it becomes as normal as possible. Okay, so homeostatic imbalances. Osteoporosis, bone resorption exceeds bone deposition, so bone becomes porous. So this may be due to aging, especially, with the loss of estrogen, perimenopausally and menopausally. Estrogen helps inhibit osteoclast activity. So when that hormone is lower than normal and start to severely diminish, the osteoclasts start to kind of go wild because nothing is kind of inhibiting them and putting a muzzle on them, so to speak, so that they start to demineralize that bone. Spongy bone, vertebrae, and femoral necks are most effective because they're very weight bearing. So they're the ones that are going to be most prone to fracture. Okay, so bone breakdown, bone resorption exceeds your ability to lay down new bone, so it becomes rather porous. Right, osteomalacia is generally a dietary insufficiency of calcium and/or vitamin D. Not everybody gets a lot of milk. Some latitudes get very little, during parts of the year, sunlight. And we know even in Florida, we get a lot of sunlight, but we still have vitamin D insufficiencies. So you need to get it in your diet as well. And that's why milk is generally vitamin D fortified, because the calcium, is in milk. It's in cow's milk, and goat's milk and things are very high in calcium. So if we fortified it with vitamin D, we have the calcium in the diet. We have the vitamin D that can be converted to calcitriol, which targets the small intestine, to absorb the calcium in the milk that you're drinking. If you have a deficiency of those things, it can lead to rickets. That's softening of the bones during childhood, and they start to bow because they're very weak. Bow and bend. Normal bone is up to the top part here. That's normal trabeculae. And then look at osteoporotic bone. Each trabeculae looks like it's moth-eaten and withered away. That's the demineralization that occurs. Okay, and that is the end of the skeletal system.